So we're very pleased to have today with us Dr. Adam Steele from Dartmouth College. Adam did his BA in neuroscience with a minor in English composition in Vassar College and graduated with honors. He did his PhD in biomedical sciences at the University of Oxford under the supervision of Professors Chris Baker and Charlotte Stagg, where he examined the functional plasticity in the human brain. He then did a postdoc at Dartmouth College with Dr. Caroline Robertson and is now a postdoc fellow at the Newcomb Institute for Computational Science and the Department of Psychology and Brain Sciences at Dartmouth College. He won multiple awards and honors, including the Innovation Award of the International Biomedical Research Alliance and the Graduate Research Award by Lincoln College in Oxford and the NIH Post-Baccalaureate IRTA Fellowship. He served as a guest editor in a special issue of Neuropsychologia and is a reviewing editor in Frontiers in Human Neuroscience. His research focuses on how humans understand and remember what they see and how these processes support spatial cognition. He's interested in the neural computations that give rise to these processes and guide our looking and movement behavior in the environment. He recently published a book chapter with Edward Silson on visual category-driven differences in memory, and also published exciting papers on his research in leading journals as Nature Communications, eLife, and J Neuroscience. So without further ado, I'd like to welcome you, Adam, to our seminar to tell us about your exciting research.
All right, thank you very much, Sharon. I really appreciate that. And thank you for inviting me to speak in the series.
It's our pleasure.
Great. So from the moment this image appears on the screen, you begin performing visual analysis. For example, you'll immediately notice the street, a big tree in the center of the image, and a fitness center. Although in general, objectively, you probably won't notice very much because this image is rather boring. But for me, when this image appears on the screen, the experience is completely different. I'm bringing online a lot of remembered information that's way beyond the visual content because I know where this place is. So when I see this image, I know that I'm in Washington, D.C. The Chevy Chase Metro is way off in the distance beyond where you can see, which I used to take every day when I would get to work. And this is actually the gym where I met my wife. So this image is a really great example of how known context really changes our perspective on scenes and changes what we interpret from the objective visual content of an image. But how is this visual content brought to bear? But what mechanistically is bringing this remembered content to bear on the visual image? The specific context that I'm interested in is the knowledge of the visual spatial environment. So the spatial layout and visual features that allow you to engage with your immediate surroundings. And this kind of information is really what we need to be aware of in order to move effectively through the world. So a lot is known about how the brain accomplishes visual analysis of scenes and also how the brain represents spatial information. With respect to scene analysis, we know that there are 3 scene selective regions of the brain, one on each of the ventral, lateral, and medial surfaces. And these areas collectively identify landmarks, determine navigable space, and derive other useful types of visual information from a scene. We also know that the hippocampus and entorhinal cortex are key regions for place memory and contain specialized cells for representing spatial information like place cells and grid cells. But how do the perceptual and spatial memory systems interact? Specifically, how does information from memory become available to the visual system in order to set up expectations about where we will move our bodies and direct our gaze? So that's the question I'll address in my talk. This talk is going to have 2 overall parts. First, I'll discuss how the brain represents perception of scenes and our memory for visuospatial context. In particular, I'm going to focus on a network of brain areas that we described in a recent paper that we think facilitate this joint representation. I'll then talk about some unpublished work about how information is conveyed between the scene perception areas and this new network of memory regions. So I'll start with how visual scenes and memory are represented in the brain. So the canonical view of how perception and memory processes exist in the brain is that they should share neural substrates. So this primarily comes from mental imagery, which is a type of explicit memory recall. So many fMRI studies have reported that mental imagery activates the same brain areas as perception of images does, including low-level areas like V1 through high-level category selective regions in ventrotemporal cortex. In fact, I would say that the classic understanding is that the neural substrates of perception and memory are shared. However, as we're kind of moving forward in the literature, we're observing mounting evidence to the contrary. Specifically, there was evidence piling up in favor of the view that memory and perception have distinct neural substrates. For complex visual scenes, this evidence was more circumstantial, and it primarily came from analyses of resting state connectivity. So these studies found that the anterior portions of PPA are more functionally connected to the anterior portions of other scene regions and to the hippocampus, suggesting that there might be a transition from perceptual to memory-based representations as you move anteriorly within these scene selected regions. However, this hadn't been shown empirically outside of resting state connectivity data. So we investigated this question explicitly, first by comparing the topography of memory and perceptual activity in the case of scenes. So for this study, we intensively mapped scene perceptual activation and what we're calling place memory activation in 14 participants to enable a fine-grained comparison. So we performed two different functional localizers. The first localizer was a scene perception localizer, and the second localizer looked for place memory preferring regions. So the scene perception localizer was kind of a very classic paradigm in visual neuroscience, where participants saw blocks of scene, face, and object images, which were presented very rapidly. And we compared bold activity during face versus scene perception blocks. The second localizer looked for place memory preferring regions. And for the place memory localizer, we had participants give us a list of 36 personally familiar people and places. So these were things like your mother or your kitchen. Then in the scanner, we showed participants these stimuli, and we asked them to vividly recall visually those places and people in as much detail as possible. And they did this for 10-second periods of time for each stimulus. And what we were doing was contrasting activity for scenes versus place vision and scene versus place memory independently in these two localizers and looking at the intersection of their activity in individual participants. So on to our results. Here's one subject's scene perception localizer. This is the ventral surface. In hot colors, you'll see the scene perception area PPA, and in cool colors, you see the face selective area FFA. Here's OPA again on the lateral surface and MPA on the medial surface. So now I'll show you where the memory activation occurred. I'll start first with the lateral surface. So again, this is OPA. So OPA is going to remain outlined in white, and I'll show you where the memory activity occurred when people were doing place versus people memory. So this is the memory, this is the activation for that place specific memory. As you can see, the activation is dramatically anterior to the perceptual activation, and there's really only a small amount of overlap between the perceptual and the memory activation. And if I flip back and forth, you can really appreciate the difference between perception and memory in OPA. So this anterior shift was present in every scene area on each cortical surface. So here's perception, and here's memory. So you can notice that dramatic anterior shift in all of the regions. And if you take a look at FFA here, what you'll see in this individual participant is that there is no real anterior shift in FFA. If anything, there's a slightly lateral shift, but the activity is very circumscribed within the overall FFA region. So this suggests that it might be a scene-specific effect. So instead of showing you all of our individual participant data, I'm going to collapse it together in these rows plots. So what you see is a center of mass of perception is the center of the rows plot, and the stem indicates the center of mass of perception. Anterior shifts are always going to be up on the slide. So the topography of responses was remarkably consistent across participants. All subjects showed an anterior shift of place memory relative to perception on the lateral surface, where there was an anterior and dorsal shift, the medial surface where there was largely an anterior and ventral shift, and on the ventral surface where the shift was pretty directly anteriorly. And there was no significant shift for faces. If anything, there was a slightly lateral shift, but no major anterior shift here. Again, suggesting this is a scene-specific effect. So we found a reliable topographic distinction between the perception and memory, between perceptual and memory processes for scenes, but do these areas actually act as distinct functional networks? So to address this question, we looked at the co-fluctuation in activity of the perception and memory areas while participants watched a movie that was meant to engage spatial processing. So this video was a kind of concatenated group of videos from YouTube, which were a kind of composite of campus tours from colleges, architectural tours, and real estate advertisements. But the important thing here is that it was all meant to give a very strong sense of space. And what we looked for was a greater correlation of activity within each network. So among the perception to perception and memory to memory areas compared to the correlation between these two networks. So what you see here is the within and between network correlations on the the within and between network correlations on the x-axis and the R values on the y-axis. So like we expected, we saw very strong within network correlation for both the scene perception and place memory networks. And this correlation was significantly lower between networks. So this confirmed that these regions do form distinct functional networks during this naturalistic movie watching task. Although I want you to note that these correlations between network are not zero. So these areas are still kind of communicating across networks. It's just significantly less than what they're doing within network. We also looked at the correlation of activity between these networks and the primary visual cortex and with the hippocampus to see whether the scene perception areas would be closer aligned with primary visual cortex and the place memory networks would be more closely aligned to the hippocampus. And that's indeed what we found. So the scene perception areas were much more strongly connected to V1 than the place memory areas were, and the place memory areas were much more strongly connected to the hippocampus. So we see this dissociation between perceptual regions and in memory regions, and we think that the fact that their correlation between networks is not zero suggests that this is a good functional bridge between the perceptual system and the memory system in the case of scenes. So, so far, we've shown a strong dissociation between these regions during memory recall, where the place memory areas seem to uniquely contribute while the scene perception areas did not. But do the place memory areas contribute to the perception of familiar scenes? So we investigated this by assessing the magnitude of activation of the place memory areas and scene perception areas when participants viewed personally familiar scene images that we downloaded from Google Maps. So these were places that the participants said that they knew from their real life, and we contrasted them with other participants' real life images. So what we're looking for, if a region contributes to processing of familiar scenes, is that there should be this familiarity boost. So here's the data from the lateral, ventral, and medial surfaces This is the activation when viewing familiar versus unfamiliar places on the y-axis. So up means more of a familiarity effect. And consistent with their involvement in memory, these place memory areas show a greater response to familiar versus unfamiliar places. That's even greater than the familiarity effect that we see in the scene perception areas. So we see that in the lateral, the ventral, and also on the medial surface. And this is a very strong effect that's present in virtually all of our participants. All right, last piece of data in this first section of the talk. So finally, all of that has been about the magnitude. Of course, it could be that these regions are just involved in the memory process, kind of recollection per se, but not actually involved in representing the information that's being recalled. And also, we've been using personally familiar stimuli. And the thing about personally familiar stimuli is you have lots of information that's coming on board, right? You have episodic associations, you have semantic associations, there's all kinds of information. And what we really want to know is whether this information that's being represented in the place memory areas is specifically visuospatial. So the way that we began to move in this direction was by turning to virtual reality. So what we did was we taught participants a set of 20 scenes in virtual reality, and then had them in the scanner, that was outside the scanner. In the scanner, we had them recall those scenes from memory, just like we did with our personally familiar places. And what we were looking for was not whether these regions were activated, which they were, but whether they were actually representing the identity of the place that was being recalled. And we found that they were. So I'm just going to show you the correlation matrices for the sake of time. And what you can see in these matrices is that there's a very strong diagonal in each of the place memory areas compared to the off diagonal, indicating that indicating that we have a significant ability to decode identity within these regions. So this suggests that not only are the place memory areas involved in memory processes, but they're actually representing the identity of the information that's being recalled. So that wraps up the first section of my talk. So hopefully I've convinced you that there is a set of brain regions that are anterior and adjacent to the scene perception areas. And these memory areas are selectively involved in the processing of familiar stimuli. They constitute their own functional network, and they're preferentially connected to the hippocampus. But what we don't know from that study is how or by what mechanism the scene perception and place memory areas are sharing information. So it's quite clear that these regions are sharing information. We can see that in their functional connectivity and also in the overlap in their general topography. But the mechanism that's underlying that communication is not clear. So I'm going to cover this in the next section of my talk. So as I just discussed, the place memory areas are always anterior and adjacent to the scene perception areas. So these areas are well poised to provide that top-down information from memory to bias scene perception and enable memory-based predictions like during natural behaviors like navigation. But what mechanisms allow information to be shared between these perceptual and mnemonic regions? So this is generally a puzzle because the memory areas are not thought, because memory regions in cortex are not thought to encode visual information per se. For example, if we look at the locations of the known retinotopic maps, we can see that the scene perception areas fall largely within these maps. On the other hand, the place memory areas are anterior to these maps. So we wouldn't necessarily consider the place memory areas visual per se, and we wouldn't expect them to use a retinotopic coding scheme. Instead, the place memory areas tend to fall on the edge of the default mode network. Unlike the visual regions, the default mode network is thought to use an abstract or amodal coding scheme rather than a retinotopic code. So this is our puzzle. How can the place memory and scene perception areas interact if they do not share common coding principles? So how do we resolve this? Well, one clue comes from a recent observation in humans and non-human primates, which has found evidence for a retinotopic code that exists broadly in cortex outside of regions that are classically considered visual. So this is a figure from one recent paper that used PRF mapping and found atypical negative population receptive fields in the default mode network, which includes the lateral parietal cortex and medial temporal lobe. So this presence of retinotopic coding, even if it is, kind of this atypical negative coding, which I'll come back to in just a few slides, that presence suggests that these high-level areas might actually use a modality-specific code after all. However, these studies have stopped at the observed presence of negative PRFs, so we don't know what functional role these negative receptive fields might play. So in this section of the talk, I'm going to describe a series of experiments where we explored this hypothesis. Specifically, we thought that the retinotopic code used by the visual system would persist in memory areas to structure the interaction between perceptual and memory systems. We focused on 3 questions. First, do the place memory areas implement a retinotopic code? Second, do the scene perception and place memory areas share visual information? And third, does the retinotopic code structure the interaction between these regions during recall? So I'll start with whether these regions implement a retinotopic code. So for this study, we had 17 participants. We performed retinotopic mapping using a scene stimulus, which flashed through a bar aperture that could traverse the screen. We analyzed our data using AFNI's PRF model, and we considered any voxels with PRFs greater than 8% variance explained to be exhibiting a retinotopic code. So on to our results. This is our group average PRF amplitude map on the lateral and ventral surfaces. So consistent with prior reports, we found that robust retinotopic coding was present throughout the brain. Very, very widespread, much more widespread than has been previously described. Here I'm showing canonical positive PRFs in hot colors and negative PRFs in cool colors. So this largely replicates prior work. So now we'll circle back to what are negative PRFs. So I'll admit that they're rather perplexing. So I'm just going to show you 2 example PRFs to give you a sense of what the responses look like and help give you an intuition of what's actually being fit here. So here's one example positive PRF from a subject's perception area, along with its corresponding time series. And this is a negative PRF from the same subject's memory area. Although these PRFs represent similar regions of space, the negative PRF shows a clear negative bold response compared to the voxel's baseline activity, but only when the stimulus appears within its receptive field. So here I've added the group level scene perception and place memory regions of interest from a set of individual participants. And interestingly, I hope you can notice that the negative PRFs really emerge within the place memory areas. So right at the boundary there of that group level lateral place memory area, you see negative PRFs begin to emerge. But because whole brain analyses can obscure the detailed location of functional areas present at the individual participant level, we turned to individually localized regions of interest to quantify this effect. So here is one individual participant's PRF amplitude map, along with their individually localized scene perception and place memory areas. The scene perception areas, which I've outlined in black dotted lines, have almost exclusively positive PRFs, and the negative PRFs really emerge within the memory areas, which are outlined in white. So this is where this plot shows the proportion of negative PRFs in each region for all subjects. So across participants, OPA contained only 3% negative PRFs, so it had virtually all positive PRFs. And this contrasts strikingly with the lateral place memory area, which contained on average about 60% negative PRFs, with some subjects having as many as 100% negative PRFs in this area. And this pattern was very consistent in the right hemisphere of OPA, as well as in both hemispheres on the ventral surface. So across the board, memory-related areas tend to have more negative PRFs than perceptual areas. So what does this mean? Well, let's say you're at the beach and you're looking at this palm tree. If the tree is not in a voxel's receptive field, there will be no change in activation. Now let's say you make an eye movement that moves this tree into a perception area PRF. This would result in a positive activation. If the perception and memory areas interact, when a positive PRF in the SPA is active, a negative PRF that receives input from this upstream positive PRF will be inhibited, resulting in a decreased bold response. So visual information is encoded in these memory areas as the result of feed-forward inhibition. So before moving on, I want to make one more quick aside about the visual properties of these anterior areas, and that's about PRF size. So the typical conception of the visual system suggests that receptive fields get larger as you move anterior in the brain. From primary visual cortex moving to the higher level areas, PRFs are getting bigger. So what we would expect is that memory areas should have bigger PRFs than perception areas, but that is not what we find. So here I'm showing the group average PRF size map, again with these group ROIs. Smaller PRFs are in hot colors. As you can see, PRFs do become larger as you go towards the scene perception areas, but then they transition back to being smaller when you move anteriorly into the memory areas. And this effect is very prominent when we quantify the effect in individual participants. So you can see the scene perception area average size in the gray bars, and the place memory area PRFs, which here I've split by positive and negative PRFs, are in the colored bars. And in both the positive and negative PRFs in the memory areas, you've got significantly smaller PRFs, which is a very interesting phenomenon that we're actively exploring and thinking about. So the answer to our first question is yes, the place memory areas have a retinotopic code. And for the remainder of the talk, I'm going to focus on negative PRFs in the memory areas because that's where we think is the most interesting interaction and distinction between the place memory and the scene perception areas is the fact that there are negative PRFs there. But I'm happy to take questions about positive PRFs if people are interested. So we next turned to whether retinotopic information structures the interaction between perceptual and memory areas. And our first step here was to look at perception, specifically whether the perception and memory areas are sharing visual information. So as we know, the scene perception areas have biased visual field representations such that OPA overrepresents the contralateral lower visual field and PPA overrepresents the contralateral upper visual field. So this is the elevation bias that I'm talking about is quantified on the right, where you can see that PPA is overrepresenting the upper visual field, OPA is overrepresenting the lower visual field. Our question is whether the biases are recapitulated in the place memory areas. And the answer is yes. So here are the visual field coverage plots for the negative PRFs in the memory areas. And what you can see quantified on the right is that these visual field biases are clearly recapitulated. The contralateral LPMA overrepresents the contralateral lower visual field, while the ventral place memory area overrepresents the contralateral upper visual field. So this is consistent with a close interaction between the negative PRFs in the place memory areas and the positive PRFs in the scene perception areas during perception. So the answer to our second question is yes. The place memory areas and scene perception areas do share visual information. So finally, we wanted to know what happens when memory is in the driver's seat. Specifically, what happens when participants engage in top-down recall, which activates the memory areas? Would the retinotopic code still structure the interaction between the perception and memory regions? So let's return to our example of the palm tree. During vision, we have positive activation in the SPAs and negative activation in the place memory areas. What should happen during bottom-up imagery? Well, we would expect the pattern to be flipped. The memory areas should be activated during recall, and their top-down inhibitory connections should cause the SPAs to deactivate. And importantly, if the retinotopic code structures their interaction, we would expect this opponent dynamic would be stronger for PRFs that are representing similar regions of visual space. So we evaluated this interaction on a trial-by-trial basis in a recall task, which was very similar to that localizer task, where participants visually recalled places that they knew for 10 seconds. We extracted activation estimates from each trial, and we looked to see how activation of the negative PRFs in the place memory areas was related to the positive PRFs in the scene perception areas. And we expected evidence for the opponent interaction that I described. On trials where place memory area activity is high, we would expect activity of the scene perception areas to be low. On trials where the memory area activity is low, we would expect the perception area activity would be relatively high. And we looked for this relationship across all trials in all participants. So these activation values are normalized to account for common signals like motion that would cause a positive relationship. So you can consider these values themselves to be arbitrary units. Again, what we're looking for is a negative relationship between the negative PRFs in the memory area on the y-axis and the positive PRFs in the perception area on the x-axis. And that's exactly what we see. When activity of the negative PRFs in LPMA is high, activation of the positive PRFs in OPA is low. And we see the same thing on the ventral surface with the ventral place memory area and with PPA. So finally, we wanted to know whether the retinotopic code structures the trial-by-trial opponent interaction that we observed. In other words, do voxels with similar PRFs that we established during visual population receptive field mapping have more correlated activity during memory recall? So to assess this, we took each OPA PRF and found its best matched negative PRF in a memory area. We then found the correlation in trial-by-trial activity between these PRFs with matched visual field locations as we did in our prior analysis. And we compared that correlation with the correlation between OPA PRFs and randomly paired negative PRFs in the memory areas. And we performed this matching and randomization procedure 1,000 times. So I'll focus on OPA first. The scatter plot is what you saw before, which is the trial-by-trial responses in all participants. So we can summarize this, this trial-by-trial response in each individual participant as their specific R value. And that's what I'm showing here. Each dot is a participant, and you can see the negative relationship between OPA and the lateral place memory area is present. Now, what we would expect is this relationship to be diminished for randomly matched PRFs. And that's exactly what we see. There's a significantly stronger negative relationship for matched PRFs compared to unmatched PRFs. And although the number of subjects is reduced because of our PRF matching criteria, the same is true for the ventral place memory area. So we have evidence for an opponent interaction during recall and during perception, as I showed with the visual field coverage. But what happens when in normal situations when we would expect the perception and memory systems to be working together? During most day-to-day activities, we're dynamically looking around places that we know very well. Would we expect there to be an opponent interaction in this case? To address this question, we ran a specific experiment investigating the perception of familiar scenes. So specifically what we did was have participants view portions of familiar scene images. In this case, we were presenting them only to the lower, we were presenting them only with the lower left and right portions of scenes from Dartmouth College, and these were all Dartmouth College students. We used the lower portions of the scenes to maximally drive OPA and the lateral place memory area, because these regions were most robustly localizable in our full set of participants. So we had there were two possible hypotheses that we were testing. On the one hand, we might expect that the perceptual and memory areas would be working in a mutually excitatory manner. For example, if they're both jointly processing a familiarity signal. This would be shown in the plot as a positive bar where all the subjects are showing this positive activation. On the other hand, if the opponent dynamic is a general principle describing these regions' interactions, we would expect to see their activity remaining negatively correlated. So this would be the case if these regions were engaged in a mutually inhibitory interaction, like predictive coding. For example, top-down inhibition from the place memory areas could come in after a familiar scene is presented. So which did we find? This is our actual data, shown again as the bar plots for individual participants' correlation coefficients. And we found that there was a significant negative correlation, and that negative correlation was specific to when the stimulus appeared in the contralateral hemifield, which is consistent with a spatially specific opponent interaction. When the stimulus appeared in the ipsilateral, so the non-preferred hemifield, this opponent interaction was significantly reduced. So this suggests that the place memory and scene perception areas exhibit an opponent interaction, and that this is quite potentially a general mechanism for their interaction. So yes, a retinotopic code structures the opponent interaction between the perception and memory areas during recall, and also during perception of familiar scenes. So to sum up today, I've shown you that visual information is encoded in the memory areas as a result of feed-forward inhibition, or this section of the talk rather. And moreover, we show that functionally paired perceptual and mnemonic areas are using this retinotopic code to structure their interactions. And that concludes the second section of my talk. So to sum up, today I've shown you that there's a unique set of brain regions anterior to the scene perception areas that are selectively engaged during recall of personally familiar places. And these areas, together with the scene perception areas, use a retinotopic coding scheme to share information. One open question is whether this retinotopically grounded opponent interaction is a general principle outside of the realm of scenes. So the place memory areas encompass only a small portion of the default mode network, which is shown here in purple. And if I once again overlay the PRF amplitude map that you saw before, you can appreciate just how widespread these negative PRFs are within the DMN. So it's possible that other functionally paired mnemonic regions in the DMN and perceptual areas in visual cortex also use this opponent retinotopic coding scheme for relating information. So the first section of the talk is published information or published in a paper in Nature Communications in 2021. The second section of the talk has actually just been posted on bioRxiv. I want to thank the whole Robertson lab, and I especially want to thank Ed Silson, who's a longtime collaborator and co-first author on the retinotopy project. And here's the QR code if you want to check it out. And with that, I'll take questions.
Thank you very much, Adam. This was really, really interesting. I'm going to ask everybody to unmute yourselves so we can give Adam a big, well, I'm clapping. And I'm opening the stage for questions. I'm happy to go first. Funny. So I have many questions, but I'm not going to bombard you with all of them at once. So first, it's just by the list of things that I've written down during your talk, I want to ask if you have any idea where EBA falls with respect to some of these areas, and whether you think that some of this may be, I mean, I know EBA may be more ventral in a way. I mean, I assume it's more ventral, but is there any, is it adjacent, or is it, far off enough?
Yes, that's a great question. So we've been looking recently at kind of general possible category specific memory areas that are akin to what we see in the scene perception areas. And it's interesting that you honed in on EBA as a possible region that has this, because what we seem to find, and I don't have this information in my slides generally, but I'll just bring up this plot here just to kind of gesture at. Can you see my mouse?
Yeah. We can. I can.
So we would find EBA kind of nestled in around this area. So
Mm-hmm.
kind of like, not necessarily ventral, but definitely, definitely ventral to where we see the scene areas. And we do, EBA is one of the regions that we do seem to have a suggestion that there might be an anterior shift during memory as well. So it doesn't appear to be the case for the fusiform body area or for the fusiform face area, or for the or for OFA. So it looks like there may be some level of anterior shift that's happening that is specific to body perception. It's unclear what that anterior shift could be, right? It could be something about action understanding or predicting actions. Like these are the kinds of things that we're very interested in unpacking as well. It does seem when I have looked at the kind of overall picture with respect to the group maps and the group PRF amplitude maps, it looks like this anterior shift is still in positive PRFs. So the shift to negative PRFs might be more specific to the scene perception and place memory areas. But yeah, I think the notion of what the action, like what actions are doing, what action understanding is, and predicting the visual consequences of human body moving in space, I think is probably something that we're going to be following up on.
Okay, brilliant.
See a
Thank
hand.
you.
Yeah,
I see Asaf has raised his hand. Asaf, do you want to have a go?
Yes, please. So great talk, Adam. A lot of questions on my end as well. I think now more on the like broad kind of implications and potential, things that made me curious. So I guess the first question regards kind of, more like causality, or why would, I guess kind of wondering, why would scenes have this memory specific system? I mean, why not other types of visual kind of stimuli? And so, how do we know that is scene specific in the sense that, I don't know if you've actually tried like just rich visual stimuli in general. So that's on the one hand. And then in terms of causality, what about the neuropsych patients? I mean, to the best of my knowledge, I don't know that there are like, scene specific amnesics that have lesions in these potential areas. And then lastly, and just a thought, and I'm sure you probably thought about that experiment yourself, if you would try to TMS these areas, what would you predict regarding, scene memory?
All right, so all three of those are really excellent questions. All things that we've been thinking a lot about. So I'll take your second question first, which is, I think about, if I remember correctly, is about the neuropsych patients, or was that your first question?
Second.
So then with respect to neuropsych patients, there are some really interesting scene specific agnosias that do occur. They're not called scenes, they're not classified as scene specific agnosias. They're actually called topographic agnosias, right? So it's people who retain the ability to recall or to visually perceive scenes, to visually identify what scenes are, but have lost their ability to use that visual information to guide their navigation, and to guide how to put their bodies in like appropriate locations in space. So there have been a few patients described in the literature with medial parietal lesions in particular, who have lost their ability to totally to do visually guided navigation. They can do or navigation based on a series of instructions, but they can't do it visually. On the lateral surface, if you get these anterior dorsal parietal lesions, people begin to have misappropriate body locations, inappropriate ways of moving their body in space. They're no longer able to kind of coordinate this, goal-directed movement effectively. And then if you have lesions anteriorly on the ventral surface, you see patients who have what they call landmark agnosia. So they're able to recognize scenes, but they're not able to recognize landmarks and use those landmarks as navigationally relevant, kind of beacons, if you will. So all of these agnosias are not like kind of classically visual. They're more these higher level integrative things that kind of require, like in a way, require this interaction between perceptual and memory processes. And so that's why one of the reasons why we think this is super interesting. So your first question, if I'm remembering, is about
Why scenes?
why scenes. So third question was TMS. TMS would be super interesting to do. We would expect similar dissociations to what we see in these what we see in the patients, but we haven't actually tried that out yet. I would love to do it, and we've got some plans, especially on the lateral surface, super accessible for TMS. The first question was why scenes, and this is because at a high level we think scenes require are a special class of stimuli where we are always maintaining things that are outside of our field of view. So we are not surprised when we turn our heads and reorient to see, a door over to my right. And so that information is always being prepared in order to make eye movements. You could argue that a similar process is undergoing, that is being used to do object permanence, but we think of scenes, because of this like existing within them, as being a somewhat special case that might be different in kind from what goes on with object permanence. But I think that's a great question, and like I said, it's one of the reasons why we're exploring these memory for other types of categories, to see whether or not this is like a task-based effect. Like, can you see these anterior shifts if you give an object in an object region, if you were doing the correct task on that object? So task demands are going to probably play a role here too. But yeah, thanks for the questions. These are great.
Thank you, Adam.
Vadim? Yeah. Vadim, sorry.
Mm-hmm.
Yeah.
First of all, thank you for excellent talk and interesting stuff. I was wondering something about terminological question. You're calling this region memory regions. Based on your experiments, how you can be sure that it's not imagery? Basically, obviously, we imagine something, we need to have it in memory. I don't know how we can imagine something without memory, but once you call it memory, so it's you directly start thinking that it's a memory system and so on, but maybe more like imagery.
Yeah, I really love that question. So we've been calling them the memory areas mostly to give us a handle on how to talk, like to give them a semantic handle so we can, begin to unpack what they are. I think naming them is a really, puts us in an odd spot. We do call them, we do think that they're involved in memory more generally on the basis of kind of the three pieces of data that I showed in the beginning, which, like you said, they are involved in imagery for sure, but they also are involved in recognition memory. So when people are just seeing either images or videos of personally familiar places, or places that we've trained them in VR, so non-personally familiar places, these regions do preferentially respond compared to what the scene perception areas do. Third, they're more connected to regions that are classically associated with memory, so regions like the hippocampus and also the default mode network, as you can see in this figure here. So on the back of those three, like more general kind of memory related functions, we and kind of results, we feel comfortable calling them memory areas, but it's been suggested that we could be calling them kind of the OPA-M or just anterior OPA. I think that those labels would also be appropriate. I'm not tied to memory areas beyond a way of distinguishing them from their perceptual counterparts.
Because when you show this evidence, dissociating them from the perceptual scene selective, it's by itself, to best of my understanding, it doesn't resolve my question regarding the imagery, because, for example, something is activated during imagery when not seeing. Okay, this is those two regions, set of regions are distinct. It's because actually I'm coming from a bit also from default mode network literature, and obviously we do know that there are regions in default network they are not exactly overlapping with the perceptual region. So with the face region completely not, but with the scene probably partly. So it's not surprising that, but the question is, if I make a paradigm in default network with some spontaneous imagery, it will also activate it. Obviously, as I said, as you said, it's memory, but maybe I don't know. Maybe it's possible somehow to manipulate the memory engagement, like if you just give a picture 2 seconds before and I ask to imagine, or whether it's a picture from your past, so it's different memory engagement, and so it's probably possible to see some.
Yeah, so I think that's a really so totally take your point. I think it's true that what we have pushed here is really an image-based task. It's because we've like the specific process that I'm very interested in studying is exactly what you're saying, you know, kind of how are we representing the actual image content as a proxy for doing this predictive processing that's being fed back potentially to the scene perception areas to guide attention or plan eye movements or do things like this. So it definitely, the way that we're thinking about memory does have a visual is a very much a visual vision first in that way. So yeah, calling them memory, like I guess that's why the place there is important, right? It's a place memory, because we do think that they're specifically involved in places, and we can rule that out very effectively by the contrast with faces and also other categories that we've tried, like objects, bodies, just very general mental imagery, and also during with perception of unfamiliar places. So we can very effectively say with confidence that it is something about places and it's something about memory generally for places, but not necessarily memory per se, like this kind of amodal, very abstract notion of memory that you're kind of referring to, I think. I did have I think one of the things that we're interested in is knowing just how widespread this notion of amodal, like whether or not these are moving even more towards an amodal representation, like a maximally amodal, where there is like, it is more of a general memory aspect, like you're saying. These regions in particular, we think are doing visuospatial, in part because of another study that we've run. Let me see if I've got an extra slide here that shows this. This will demonstrate it a little bit. So in this study, we also used VR. We showed participants images, panoramas, and streets. So it's parametrically modulating the amount of visual information that they experienced. And what we found is that the place memory areas parametrically vary with the amount of visuospatial information that's known. So we think that that's potentially definitely an indication that they don't need episodic information, because these are VR trained scenes that the participants are experiencing only a handful of times. The only difference among them, they're visually matched, so the only difference among them is what the extent of visuospatial knowledge is. And the scene perception areas do not show this modulation. They actually don't show, OPA and PPA don't show any modulation above baseline.
What is the difference between panorama and street?
So panorama is, they're wearing a VR headset. Image is just 45 degrees. Panorama is 270 degrees. And then street is 3 photospheres, so they can advance by pushing a button. And once they get to the final photosphere, they can actually look down a corner that they couldn't see from the first photosphere. So it's like it's like a image, panorama, and then panorama plus, if you want to think of it that way.
Another I'm sorry for interrupting you, Adam, but another
Oh, no
relevant
problem.
concept in that particular context is the notion of navigational affordances. And you
Yes.
can think about reachability and explorability, and there's work from Russell Epstein, and we did some work on that. So you can really see how this scene selective regions are also engaged in coding the potential for action, which is in scenes would be exploration. Now, if you relate that to your story on memory, and that would be kind of engaging your system for computing those, large scale geometrical constraints, that would really combine all these kind of works together.
Exactly. So that's exactly the framework that we're thinking about these place memory areas in. So OPA is a great example where OPA itself is probably involved in path finding, finding navigable paths through a scene. There's great evidence from a number of different labs showing that. We think that the place memory area on the lateral surface might be involved in representing kind of what's next, right? Because as you move through that path in a scene, you're going to continue seeing other paths. You need to plan all of that route, even beyond what you can see. And we think that that might be what the place memory area is doing. It's kind of acting as a way station or translation point between the goal-directed processes that are happening in prefrontal cortex, prefrontal hippocampal interactions, and also the visual interactions that are finding what's immediately in front of you. So that's exactly the kind of framework that we're hoping to build towards as we're moving towards, as we're trying to understand basically how these relate to the scene perception areas. So yeah, it's a fantastic point.
I also, if it's okay to, I'd like to follow up, and actually it relates to both of the issues that both Vadim and also Asaf have raised. So a few questions. This is also relating to what Asaf has said, not time-wise, but if you examine the distance-wise of the stimuli versus the body. So would you find that scenes or stimuli that appear to be closer to the observer, let's say personal, reachable space versus things that are more distant, would you find a difference? I also, with respect to the patients that you discussed, so first of all, it would be interesting to see if there are any reports in the literature that show that these lesions lead to neglect, whether they do or they don't. And to also navigational aspects, which parietal is sometimes related to navigational skills lost. And there's also, and that would also lead me to the right-left differences, whether you find any differences between right and left hemispheres with respect to these areas. And okay, you don't have to answer all the, just telling you things I had in mind.
Yeah.
So there is an overlapping area, and each of these also have like the DMN and there's the area that's overlapping between, let's say, the memory and the DMN, and the memory and the perception areas, and there are areas which don't overlap. And do you see if you restrict your analysis to the areas that show overlap versus the portions that don't overlap, either within the DMN or within the perception network? So do you see any difference if you just examine these portions separately? And okay, I'm just there's also
I can't keep all of these in my head, so I'm just going to start.
No, I have it written down, and I'm also going to email it to you.
Okay, great.
So on top of the patients losing their ability to navigate, it's known that, there are a few reports, I can send them to you, that were actually, it's like, following the trajectory that was found with prosopagnosia, that it was thought that it's only an acquired issue, and then it was found to be a developmental issue. So now it's also known that there are developmental deficits, people who are actually finding it really difficult to navigate, even within the very familiar environment. They've been assessed. And so it would be interesting to see whether they have normative activation within these regions. because it could be that this is, this is necessary, but not sufficient. So it would be interesting to see if these regions show normative activation patterns, and also with respect to that, it would be interesting to see what happens if you TMS perhaps different portions. I was also thinking of the drawing paradigm that Wilma Bainbridge has done, versus the familiarity, whether, TMS would maybe interfere with drawing, and at what temporal latency from visual presentation, and whether maybe not, or recognition recall, recognition memory, but maybe drawing, or vice versa. So I just, and another thing, Marty Serino has shown that, you see that this is fascinating, yeah. So he has shown some kind of, so it's not retinotopic map, but he has, he did this thing with eye tracking and, with the air paths. So that's way back where he found basically spatial organization that is not really only retinotopic based, but rather head based or body based or combination. And I'm wondering if what you are calling PRF is actually really related to retinotopy, which is usually what we relate to, or maybe it's something about the space that is present there, and it projects to retinotopic space, but it's actually not retinotopy, but it's more about the spatial layout in some way. Okay. Yeah. I bombarded you with all of these.
So I'll take them in reverse order as I remember.
Yeah,
them.
you don't have to address
So
all
the
of them.
first,
really.
No.
Yeah.
I think your last point on reference frames is actually a really nice, is actually a really interesting point that we're thinking a lot about. So, there is in parietal cortex in particular, lots of different reference frames that are all kind of present and somewhat mixed together. And that's because I'm kind of, as related to some of your earlier points, it's like parietal cortex is involved in a ton of different things, from moving the body in space to moving the hand to grasp objects. And so you have, body-based cues, retinotopic-based cues, kind of hand-oriented cues. You have all these different reference frames that are all coming into play. And with this with PRF mapping, we are very specifically aligning all of those reference frames. And so in a way, we can't, as you're pointing out in these data, we can't disambiguate that. But we are thinking about running either a project like with TMS, or a functional imaging project that would dissociate these things either by changing eye position relative to the head,
or you can also do reaching.
Some other manipulations like this, or reaching, exactly. So I think we are definitely interested in understanding whether this is truly a retinotopic reference frame. That being said, I do think that a retinotopic reference frame in the context of this information sharing with visual cortex, a retinotopic reference frame would be extremely convenient to maintain at some level, even alongside these other reference frames that may also be present. So what we're kind of thinking is that it may be both a combination of the tasks that we're having people do, the demands that the system has, and also the attention that the person is placing on this. What informations kind of rise to being most what reference frames rise to being most prominent at a given time. So we've definitely loaded our die here to be able to find retinotopy. I wouldn't necessarily stake myself on saying that's the only reference frame that's present in these areas, but I do think that it is one among many. So I'm trying to remember some of your. Egocentric.
Yeah.
Oh,
right or left differences.
We do see some recognition effects that are generally strong, memory related effects that are generally stronger in the left. But the magnitude of the difference doesn't seem to vary that much, which is why all of the statistics that I've been reporting have been collapsed across hemisphere. Because in general, we don't see, even though we see absolute magnitude differences, the different scores and the effects that we're talking about, you know, the anterior shiftedness and things like this, that doesn't seem to vary. And we can equally well show our data in both. But yeah, we've been keenly aware of that because of these recall related effects. It is one of the things that makes doing position specific imagery with scenes somewhat challenging. So we did try, we did have a couple participants do some behavioral work where we were trying to do position specific scene imagery. You know, like imagine the lower, like you're standing in your kitchen and imagine the lower left-hand corner of the kitchen. And it's very unintuitive for people to do. It's interesting that people can't do that for scenes, but they seem to be able to do that very well for objects. You know, you can imagine a tea kettle in the lower right or the lower left, but you can't imagine, but people have a really hard time selectively imagining portions of scenes. I think we you know, whether or not this is like a confound or whether or not this is a very interesting like aspect of the imagery and place understanding phenomenon is kind of like two sides of the same coin. I personally think it's like related to the way that we actually do imagery of scenes is related to the mental or the neurological mechanisms that are at play here, where what people often say they were doing in this behavioral pilot is not placing a scene there. It was, I'm imagining I'm in the place and then I direct my attention or plan an eye movement to that area and everything else kind of fades. And I think that's really interesting. I think that says a lot about the mechanisms that might be at play here. Eye movements are going to be, you know, planning eye movements is one of the things that we think might be related to the types of processes that we're studying here. But yeah, I think all those questions are really great. I wish I could answer all of them.
No, but
and I'm looking forward to it. I'm looking forward to answering all of them, I should say.
Yeah, you can write a lot of grants with all these, well, and many other.
Egocentric, your question about egocentric distance is one that I think is absolutely fantastic. Like you think that the amount of distance that you can plan things would increase, maybe you selectively do some of this route planning that I've been arguing is happening on the lateral surface. Yeah, these are all as you said, excellent grants.
Yeah, maybe there's some trajectory, you know, from, you know, very personal to extra personal space. I mean, I don't know, but you know, it could be that there is some, you would maybe, you know, if you examine that parametrically, maybe you'll see some, you know, parametric change in the activation.
Yeah, I love the idea of trying to dissociate this action space, like the actions that we perform in these different spaces, where in these very personal space, you might not expect the kind of scene selective regions or these regions, if they're doing navigation planning, to be as involved, right? Or as active compared to if you were doing something like route planning in very extra personal space. I think that would be a really interesting dissociation, and I would suspect that it would happen on the lateral surface.
Yeah, but it could.
So that's really cool. I think that's a great idea.
Yeah, it could actually also be that, you know, I mean, my thought about dorsal in general, and I see this as more like dorsal-ish, is that it has to do with here and now of our being. So that's, you know, time-wise, it's, you know, and the ventral aspects are more later, kind of like. So if this is here and now, it's also the closer aspects. So I would expect, you know, you're talking about, you know, route planning, but I would expect that it's the closer portions of the route that, you know, that our body is going to interact with just now or just a bit later. You know, the route planning, I don't know, maybe I'm completely off, but I would assume it would be more, you know, medial, ventral, kind of like that. But, you know, it's data always surprises us. So whatever my thoughts are.
Exactly. Exactly. We never thought that negative PRFs were going to exist in these regions, but here we are, like, you know, out championing that. I think like you're saying, this is an empirical question that's definitely worth going after.
So Adam, I really want to thank you a lot for this exciting, stimulating talk. It was really a pleasure having you here. And so thank you for joining us also on a very short notice. It's a blessing that we were fortunate to have you. And next week we're going to have Linda Smith with us. And we'll advertise that. But thank you very much. And have a great week. Asaf said, you know, in the chat, why am I not opening it?
Yeah,
Sorry,
left.
have to run to
Left
another
on.
meeting. But great talk. He said, great talk.
Thank you.
Yeah, thank
yes.
you. Very, very interesting.
Yeah.
It was nice to meet you.
Yes.
Thank you. Very nice meeting
Yeah,
you
yeah,
too. And I
yeah.
really appreciate the invitation to speak to you.
Yeah,
This
we're
has
very
been
happy
fantastic.
to be.
Bye-bye.
Yes. Thank you, Adam.
Bye.
Bye.
Ciao. Have a nice day.
Bye. Bye.
You too.